My name is Susan Hannon, and I'm the president of Salt Spring Island Conservancy. Before we begin tonight's webinar, I want to acknowledge the unceded territory and ancestors of Coast Salish First Nations of Salt Spring Island and the surrounding areas who continue to use and steward the lands and waters of the Salish Sea. These include the Cowichan, Penelakut, Paquachan, Malahat, Sayout, Sartlip, and Sacom nations. So the Salt Spring Island Conservancy has been conserving our island nature since 1995. We currently protect almost 2,500 acres on Salt Spring through our 8 nature reserves, 17 covenants, and 78 stewardship agreements. We also conduct educational events and workshops and coordinate the stewards and training program for school children. We always welcome new members and donations. So if you wish to join or donate, please go to saltspringconservancy.ca and Deborah will type that into the chat box. So this fall, the Conservancy is celebrating forests with 3 natural history webinars on what we call the denizens of the forest. And those are mushrooms, owls, and amphibians. So they'll be coming up over the next couple of months. And we'll be co-hosting these with the Salt Spring Naturalists. We have been collaborating with them on educational events, including last December's popular winter bird webinar, which some of you may remember. And Deborah, is David here? Or David, are you here? Just speak up if you are.
I am. I've just got here. The ferry was an hour late today.
Oh,
can you hear me?
Yes. So too bad we can't see your picture because I wanted people to see what you looked like. But did you want to say a sentence or two about the naturalists? Or
thank you very much, and I'm really excited to hear Andy tonight. And thank you to the Conservancy for inviting us to join you in offering this great program. I will say that many people don't know much about the Salt Spring Naturalists. And I really want to say that it's a new group forming right now. Look for our launch in the spring. I think it's going to be really neat to have a group that does a lot of what the Conservancy does, but also tries to offer a lot more natural history for families. So we'll be seeing that. But thank you very much, and really looking forward to hearing Andy and seeing him again. Thanks.
Okay, thank you, David. I just also wanted to mention on the theme of forests, our fall edition of our newsletter, The Acorn, will be devoted to forests. And we're also co-hosting a panel with Transition Salt Spring on forest, fire, and water. 2 people won copies of the mushroom book that Andy has recently published, and Deborah will be contacting you by email so you can pick up your copy of the book. Now to the main event. So I'm very excited to introduce Andy Mckinnon. Andy is a well-known naturalist, forest ecologist, and mycologist, and since 2014, a politician. He is a councillor for the district of Myckinnon. He worked for the BC Forest Service for 3 decades until his retirement in 2015. His work focused on ecosystem classification and mapping, ecology of old growth and effects on climate change, and BC's native plants and fungi. Andy has also been involved in defining and implementing ecosystem-based management in Haida Gwaii and the Great Bear Rainforest. He is a co-author of 6 best-selling books about plants of Western North America and was awarded an honorary Doctor of Science degree by Simon Fraser University in 2013. I first got to know about Andy when I first moved to Salt Spring Island 11 years ago, and I looked around me and realized I needed to know a lot more about the plants. And so I bought this Bible here, Plants of Coastal British Columbia, and you can see based on how dog-eared it is, how much I've been using it over the years.
Excellent.
He recently published a new book called Mushrooms of British Columbia, co-authored by Kem Luther. He told me just before this started that it's become this week BC's #1 best-selling book. So that's fairly rare for a natural history book. So congratulations. And one of the better reviews that I read about Andy's book on mushrooms said it was the new book is a love letter to BC's bounty of mushrooms. So tonight he will be speaking about fungi and forest ecology. So take it away, Andy.
Thank you very much, Susan, and thank you very much for the invitation, and thanks to everybody for coming out tonight. I had a great time doing a webinar, what was it, 8 months ago or so, Susan, for the Conservancy with Richard Hebda on forests and forestry. So it's amazing that I was invited back, but we'll take it for what it is. And yeah, I have worked with plants for many, many years, but I actually did my master's in mushrooms or on mushrooms in 1982, way back when. And so tonight I'm going to talk about fungi and forest ecology because Susan told me to. And here are some pretty pictures to illustrate a few concepts. I think the general idea tonight is that I'm to talk for about 45 minutes, which will take me just till about 8 o'clock or so, and we'll leave lots of time for questions and discussions and so forth. Can people see that all right? Okay. I thought maybe I'd start with a quick background on fungi because a lot of people are not familiar with this fantastic kingdom of organisms. So a couple of family trees here. People divide up life in a lot of different ways. These branching tree or shrub diagrams are intended to show evolutionary relationships over hundreds of millions of years. A general distinction is between the eukaryotes, which are generally big things, though some of them are quite small, that have their genetic material in a nucleus in the cell, and the eubacteria, things we might think of as bacteria, and the archaea, which are a group of extremophiles that have some characteristics in common with bacteria that are found in very hot water and very salty water and things like that, sometimes called archaeobacteria, which lack that nucleus in the cell. And among the eukaryotes, and I've always thought this was quite funny, there are different kingdoms like plants and animals and fungi. I'm old enough that when I was young, fungi were taught as being a kind of a degraded plant. And they're really quite different and have their own kingdom now. One of the things that always amazes me is that when you're looking at eukaryotes, there are plants, animals, fungi, and 23 other kingdoms. Can anybody name one? I'll leave that with you. If we look in a little more detail at some of these trees, we find, don't worry about all the names here, all the details. The plants up in the top left there in the green area, and a group called the opisthocons, which have almost all of them a very distinctive flagellum at some point, or whippy tail at some point in their life. And you can see the red box around animals down there in the opisthocons in the lower right. Can everybody see that red box? Yeah? Okay. And if we zoom right in on the opisthocons, we can see where the fungi are and where the animals are. And what that shows you is just about the closest relatives that animals have on Earth are fungi. And just about the closest relatives that fungi have on Earth are animals. In just about any way you can think, fungi are much more closely related to animals than they are to plants. Evolutionarily, biochemically, physiologically, reproductively, there are a lot of similarities between fungi and animals, and not a lot between fungi and plants. And what that means from a practical point of view is if you see a mushroom growing in the yard outside your house, that mushroom is much more closely related to you than it is to any plant. So what are fungi? Fungi are eukaryotic. They have these nuclei in the cells that contain their genetic material, you remember? They're heterotrophic. That means they can't make their own food. There aren't a lot of organisms on Earth that make their own food. Plants do, cyanobacteria do through photosynthesis, and everything else that lives on Earth depends upon them for their nutrition. And so fungi have to find lots of different ways of getting food, and that's going to form a central organizing principle of my talk tonight. And they're absorbent, so they will eat other organisms. They don't have a mouth or a digestive tract. They'll digest them outside the fungal body, and then they'll slurp them up. The fungi can be like amoebae, little single-celled organisms moving around. They can be unicellular and not moving around, in which case we call them yeast. Or the vast majority of fungi, and certainly all the ones that are large enough for us to see, are filamentous. They're made of, if you look at the picture on the lower left there, they're made of microscopic filaments. One of those is called a hypha. More than one is called hyphae. And a whole mass of them together is called a mycelium and forms the fungal body. Those cells, that's under 1,000-time microscope, those cells are about 1/20th the width of a human hair. So they're microscopic. You can't see them unless you've got a very good microscope, but all larger fungi are made out of them. Fungi generally don't move around much. The cell walls of a lot of fungi contain chitin, which is something that plants don't do, but a lot of animals, including an awful lot of insects, do. And most fungi reproduce by various sorts of spores, and there's a bunch of them illustrated in the lower right-hand corner there. Everybody listening to the seminar tonight has tens of thousands of fungal spores all over them. You just do. It's the way of life. The air is full of fungal spores. Every breath you take, you draw in fungal spores. They are ubiquitous. All mushrooms are fungi, but not all fungi are mushrooms. So if fungi make structures that are large enough to be seen by us without a microscope, they're oftentimes referred to as mushrooms. And some people will reserve mushrooms for being things that have a cap and a stalk and gills underneath. And others, including myself, will include in the broader concept of mushrooms things that look like blobs of jelly or little birds' nests or brackets coming out the side of a tree. I would call all those mushrooms, but you can decide for yourself. You're all consenting adults. And so all of these things we call mushrooms are fungi, but not all fungi are mushrooms. In fact, most fungi, the vast majority of fungi globally, don't form structures that are large enough for us to see without a microscope. Although we can occasionally see them en masse. They're the things that turn your cheese blue if you leave it out of the fridge for too long, or turn your cheese blue if you decide you want to make blue cheese. On the top left there is, I believe, a Roquefort cheese, and on the right is a microscopic photograph of Penicillium roquefortii. Penicillium is, of course, one of the most famous genera of microscopic fungi, yielding the first modern antibiotic, penicillin. All of the penicillium species look like the photo on the right there. And there are a number of species with names like Penicillium roquefortii, Penicillium gorgonzolii, Penicillium camembertii, all of which have their specialized purposes. I'm sure you can guess. And of course, yeasts allow us to bake and brew. And I'm not sure how many people at tonight's seminar would like to live in a world without baking and brewing. Those are single unicellular fungi. All right, so fungi are incredibly diverse and extraordinarily poorly known compared with just about any other group of organisms you could think about. I have lots of friends who are birders, and one of them once told me that the number of bird species on the globe was, oh, I can't remember, it was birds, but it was something, let's say it was somewhere between 7,850 and 7,900. It was something like that, the number of bird species globally. The number of fungal species globally is believed by best estimates to be somewhere between 1.5 million and 30 million. And it could be more than that, but that's best guess right now. So it's impossible to say how many fungal species there are globally within an order of magnitude. So as a group, compared with some other groups that people here may be familiar with, they're rather poorly known. There's a publication called Macrofungus Species of British Columbia. For the purposes of tonight's talk, you can think of macrofungus or macrofungi as being the same or equivalent to mushrooms. They're the ones you can see. So BC has more than 3,400 recorded species of macrofungi or mushrooms. And that's more than we have of plants or of most groups of animals until you start including the insects. So the mushrooms are incredibly diverse in British Columbia compared with other groups of organisms as well. If I was to guess, we have more than 3,400 recorded. If I was to guess how many macrofungus species we have in British Columbia, I think I'd guess somewhere around 10,000. Okay, and that's just an example of how poorly known the group is. There are certainly lots and lots and lots of mushroom species on Salt Spring Island that are new to science, without a doubt. And again, it's extraordinary. If the low estimate of fungi globally of about 1.5 million species or so, then we have described much less than 1% of them. If it's more like 15 million species, then scientists have described closer to 0.001% of the species that actually even have a name. So it's not a well-known group, let's say. Why are there so many species of fungi in British Columbia? For the same reason that BC has more species of bats or birds or any other group you could think of than any other province or territory. And it has to do with climatic diversity and diversity of ecosystems. This is the map of the biogeoclimatic zones of British Columbia. You're one of those little orange dots in the lower left-hand corner, the orange being the coastal Douglas fir zone. You want to learn more about these mushrooms? There's a new book out on mushrooms of British Columbia. The third book on mushrooms of British Columbia in the Royal BC Museum Handbook series. The first two came out in 1946 and 1964. So about time for a new one. Most of the mushrooms that you find in the woods, almost all of the things you'd think of as mushrooms are basidiomycetes. Just a few more technical terms, and then we'll leap into the forest ecology. Here's an example of a basidiomycete, the short-stemmed russula. Like any other group of organisms on Earth, fungi are divided up by how they reproduce sexually. It's not necessarily because biologists are obsessed with sex, though some are, but more because sex is believed to be evolutionarily conservative, that a lot of other things about you can change over evolutionary time, but if you start monkeying around with the way you have sex, you're not likely to last too many more generations. So basidiomycetes, if you look at the gills microscopically, they have basidia, these club-shaped structures with 4 points on them that make the spores, the basidiospores, on top of them, and there are hundreds of thousands of those on even one mushroom. And if you're looking at mushrooms or things with veins underneath or teeth underneath, boletes, conchs on trees, jelly fungi, bird's nest fungi, they're all basidiomycetes. The other major group is the ascomycetes that reproduce sexually in a different fashion. You may see some cup fungi or morels around, they're ascomycetes. So if you look again microscopically on the surface, you find these elongated sacs, each with 8 spores in it. These are the sexual spores. There are, in a lot of these species, asexual spores as well. And these are blown out the end of the sacs into the air currents and wafted away in the air currents. Here in an example from a basidiomycete, I just want to make one more point here about life cycles. And that is that the germinating spores form these thin tubes, the hyphae that form the mycelium. You can see them on the left there. And if they find a compatible mating type, then they can get together and make a mycelium together. They have a really different way of doing that, but it doesn't bear too much on tonight's topic. So we'll get into it another time when let's see if we can make a date sometime in the future for me to come over and talk about mushrooms, and then we'll go on a mushroom walk. Okay, maybe have a big mushroom feast at the end. We'll do that as something with the Conservancy. And so the mycelium, these microscopic threads in the soil or in the decaying wood, are there year round. And that's the body of the fungus, and we only ever know it's there when it decides it's time to have sex and make its sexual spores. And that's what a mushroom is. That's what a conch is. That's what a jelly fungus is. That's what a bolete or a truffle is. It's what the mushroom does to make its sexual spores. It's what a puffball is. Right? So the fungi that make all of these structures are in the substrate, they're in the soil, they're in the trees, they're in your compost heap or your front lawn year round. And that's why if there's a mushroom you're particularly fond of, you can come back year after year and find it. It's because the fungus is there 365 days of the year, and we only know it's there when it makes these fruiting structures. All right. So I promised that I would talk a bit about how these mushrooms are deriving their nutrition. This is one way of organizing information about forest ecology. And so I'm going to talk about these fungi that produce mushrooms that we know about and some that don't, as saprobes, organisms that derive their nutrition from dead hosts or decaying organic matter, parasites or pathogens, and these derive their nutrition from a live host, sometimes you. Carnivores. There are fungi that are carnivores, believe it or not. And then I'm going to talk quite a bit about fungi that are symbionts. That is, they derive their nutrition from plant partners that they live with. And there's all kinds of different arrangements of that. Let's start with saprobes. And a lot of people don't think too much about these decomposers in the forest or in the fields, or any ecosystem you can think of has fungi decaying organic material in it. They work with mesofauna, small insects and bacteria to break down the material that falls to the forest floor in this case. They include important medicinal species like the turkey tail on the top right. I know some friends on Salt Spring who gather turkey tail and use it medicinally. And really common and abundant species like the orange peel fungus, which seems to delight in nothing more than growing by the side of a trail. And sometimes I feel that these decomposer organisms, these saprobes, don't get the respect they deserve. But there was one paper that calculated that if we didn't have these organisms and their pals among the insects and the bacteria breaking down the material that falls on the forest floor, that it wouldn't take more than a couple of decades before the material that falls to the forest floor was as tall as the trees themselves, and the whole system would shut down. So they're critically essential to recycle organic material and help put it back into the soil for another generation of plants and other organisms to grow in. Just about every small mushroom that you find, there are a few exceptions, but just about every small mushroom, like the bleeding Mycena on the top right here, that you see in the forest is the fruiting body of a fungus that's a saprobe, or a fungus that's decomposing material on the forest floor. It's decaying something. The large mushrooms are something else. We'll get to that. But just about every small mushroom you see in the forest is the fruiting body of the decomposer fungus. There's a few exceptions, but as a general rule, that's a good way to classify them. And here's a very nice picture of the spirit gummy bear, which is very common on Salt Spring Island. We divided up the fungi into different groups in the book, as most books do, including jelly fungi, which are gelatinous, and tooth fungi, which have teeth underneath instead of gills. And it's really difficult to decide with the spirit gummy bear because it's a gelatinous fungus with teeth underneath. So you have to choose. In some places it's called jelly tooth. In others it's called cat's tongue, which is a name I find particularly unappealing, but then I'm not a cat guy, because the teeth, if you rub them against your cheek, they feel like the tongue of a cat rubbing against your cheek. I shudder. But we do have a made in BC common name, in fact, invented by someone giving you a seminar tonight, called the spirit gummy bear, which is what it's called in the new book, because it really is just like a little gummy bear. But if it's like any gummy bear, it's white. So it's clearly the spirit bear, BC's provincial, is it the provincial mammal or the provincial carnivore? I can never recall. Anyway, decomposers, saprobes, there's lots of them. And we think of the autumn as being the fungus season, but an awful lot of the saprobes, including a lot of the small mushrooms, will fruit any month of the year when there's sufficient moisture. Oh, that's kind of dark. Okay, that's a very dark picture of a red-belted conch. Sorry about that. And I've included it because it's both a saprobe and a parasite. So we'll deal with it in parasite, and hopefully it won't be quite so dark anymore. A lot of fungi are parasites. That is, they derive their nutrition from living hosts. If people here tonight want to have nightmares, I highly recommend going to YouTube after the talk and looking up zombie ants, if you haven't already done that. Zombie ants, there's a group of parasites called cordyceps that parasitize a lot of different insects. And in BC, they also parasitize some truffles. In BC, we find them actually growing on 10-lined beetles on the dunes outside Tofino and Ucluelet. In the tropics, they specialize in being parasites of ants, and they take over the ant and cause the ant to do very un-ant-like things. They cause it to walk away from the colony all on its own, to climb up to a place that's high and dry, usually on the underside of a leaf. All the while, the ant moves at the direction of the fungal parasite. It gets underneath the leaf. It locks its mandibles on the underside of the leaf, and when it's securely in place, then the fungus finishes killing it and fruits through its head and distributes its spores on all of its ant pals down below, and the cycle repeats. That's why they're called zombie ants, because they're no longer in control of their movements. So have a look. If you really want to have a nightmare tonight, I recommend zombie ants, or maybe hold on to it until sometime a little closer to Halloween. A really common rust on Salt Spring. So rust is there are hundreds of species of rusts. They're fungal parasites of other plants, and one of the most common and obvious ones on Salt Spring is the Fir Blueberry Rust. Rusts can have these incredibly complex life cycles. The Fir Blueberry Rust makes 5 different types of spores. I think 2 of them on Salt Spring on Grand Fir, and 3 of them on a species of Vaccinium, most commonly on evergreen huckleberry, or Cinnamocha. And what it does when it grows systematically through the Vaccinium or the blueberry or huckleberry species is it turns the stems into something that looks more like cocktail sausages than stems, where they become really fat with very few leaves. And that's just the fungus directing this disorganized growth of the Vaccinium species in order to make more surface area for the fungi to fruit on. So that's if you're wandering the trails on Salt Spring, watch for Vaccinium species that look like the top there. You're seeing a Fir Blueberry Rust. Certainly a lot of people have seen powdery mildews, just looks like powder around southwestern British Columbia, probably most commonly on bigleaf maple. And you can see it here on the maple. And of course, there are fungal parasites of people, like ringworm, which is not a worm at all, but a fungal parasite. So, oh, yikes, that is one terrible slide. This is, I included this one slide twice, and it's a crummy slide twice, unfortunately. But you'll recognize, I think, the most common conch on Salt Spring Island, the Red Belt Conch, just to show that the categories that we assign things in the natural world, the organisms oftentimes don't fit neatly into them. So the Red Belt Conch is a good example. Remember, these are just the fruiting bodies. The fungus is inside the tree, rotting the heartwood, eating the heartwood. It will infect the live tree, at which point I guess we'd call it a parasite. But after it's killed the live tree, it continues to eat the heartwood of the dead tree, in which case I guess we'd call it a saprobe. And it really doesn't matter to the red-belted conch. In fact, if you really want to generate a debate, consider that the heartwood in the center of a conifer tree is actually dead. So if you are a red-belted conch rotting the dead heartwood of a living conifer, are you a parasite or a saprobe? Doesn't matter. One of our favorite parasites is an Ascomycy parasite that parasitizes the short-footed russula or short-stemmed russula, which is a very common Russula species, very common species of mushroom on southern Vancouver Island and the southern Gulf Islands. And when it gets parasitized by this Ascomycy, the surface, which previously was white gills, gets turned into an orange pimply surface. And that parasitized short-stemmed Russula is what we call a lobster mushroom. So if we didn't have that parasite to transform physically and also gastronomically, the short-stemmed Russula, we'd never have lobster mushrooms. It transforms it from a rather a blah tasting mushroom to something that's quite delicious. There were no doubt people here tonight who have eaten lobster mushrooms. There's a lobster mushroom hand. That's just because we're close to Halloween. That bears no relation to anything. They're entirely gratuitous. There are carnivores. There are fungi which are carnivores, believe it or not. Here are some carnivores. And here are some other carnivores. Boy, a few of these photos aren't turning off. That's a really nice photo of oyster mushroom in the top right there. But oyster mushroom and witch's butter are a couple of fungi that are also carnivores. Not the fruiting bodies, but the fungus itself. So if we look at them, the oyster mushroom in the bottom right there, and we look at the filaments that make it up, right? So remember, the fungi consist of these microscopic filaments, like at the bottom left. If you were to crush an oyster mushroom on a slide, you would see that it's made up of all these little filaments. And really how all these filaments in the wood or whatever the substrate is get together to make oyster mushrooms is one of the most incredible and poorly understood examples of biological knitting in the entire world. But if you look at the microscopic filaments that make up the oyster mushroom, and in this case, the microscopic filaments on the lower left are growing in the wood, they form droplets of a substance called ostriatin, which parasitizes very small organisms so that the fungus can then secrete enzymes and eat them. Important for vegetarians to remember that the oyster mushrooms are eating meat. Okay, just dietary advice. There are cool microscopic fungi, like in the top left there, Arthrobotrys, that makes these little loops that nematodes get caught in often enough to feed Arthrobotrys. And as soon as the nematode gets its neck through one of these little loops, the loop constricts really quickly. Or Zoophagus, which has adhesive pegs that this poor little rotifer has got trapped on in the top right. So there are some fungi that are out there in the woods hunting. They are predators. Symbionts. I've subtitled this Cool Plants and Their Fungal Friends, and I'm going to talk about lichens, mycorrhizae, and mycoheterotrophs. If you're unfamiliar with some of these words, I hope you will be will not be at the end of my presentation, and if you are, then please ask in the questions afterwards. Before we get into those three, let me just back up for a second. So you remember plants can make their own food and fungi can't. And so plants and fungi over 400 million or more years have entered into all kinds of different arrangements that allow the plants to photosynthesize and produce carbohydrates that they share with their fungal buddies, and the fungi provide something in return to the plants, and the nature of the arrangement is different at different times. But because I love plants and because I love fungi, I'm particularly enchanted by the relationships they form together. I'm going to touch on these three. I don't want you to think that these three are the only three ways that plants and fungi form partnerships. And a lot of them we don't know about, we're just learning about. Here's a photo of Western yew. It should say Taxus brevifolia. Forget that it says Taxus baccata, but I was thinking of of Taxus baccata, the European yew, because there's been a lot more research done on European yew than on Western yew. In the early 1990s, I think it was, there was a bit of a gold rush for the bark of Western yew in British Columbia. Systematic studies of a number of different plants in the United States had revealed that there were substances that you could derive from Western yew that were effective in combating certain types of cancer, and most specifically useful in treating ovarian and breast cancers, and are still used to this day as chemotherapeutic agents. The only place to obtain them at the time was from the bark of Western yew trees. And so Bristol Myers Squibbs in the United States started offering large amounts of money for Western yew bark. Of course, when you remove the bark from the tree dies. And so a lot of people, well, not a lot, but a small number of people got fairly wealthy harvesting yew bark where Western yew grows in British Columbia in the interior wet belt and on the coast, and managed to wipe the species out in some of those areas. And it's still slowly returning to those areas. It was subsequently figured out how to semi-synthesize it, and the gold rush ended. And so Western yew's been making a little bit of a comeback in those areas since. But there was a very cool paper published in 2015 called Isolation, Identification, and Ecology of Growth and Taxol Production by an Endophytic strain of Paraconiothyrium variabile from English yew trees. That's why I was thinking of Taxus baccata. And from that paper, of the 242 endophytic fungi isolated from surface sterilized yew tree twig segments, only 2 species produced Taxol in a defined liquid medium. Okay, now, a few words here. Endophytic fungi. Turns out people knew that some grasses have known for 100 years that some grasses had fungi growing inside the leaves of the grasses without causing any kind of symptoms. They weren't parasites or anything like that. They just seemed to be living there. Turns out, subsequently, every single plant on Earth that anyone's ever looked at has fungi growing asymptotically without symptoms inside the leaves, and sometimes lots of different species in one plant. Here they have 242 different, well, they're not sure if they're species, but different genetic units that they've managed to isolate. And 2 of them that they were able to identify actually produce Taxol. Turns out it's not the Western yew that's producing the Taxol, or it's not the European yew in this case, but they've also found Taxol producing fungi living as endophytes in Western yew as well. It's actually fungi living inside the leaves of and twigs of the Western yew, of the European yew and Western yew. What else are they doing in there? What are they doing for the plant in return from, presumably they're getting nutrition from the plant? What are they giving back? Are they making it less appealing to animals that might otherwise browse the trees? Why do the yew trees need the Taxol, for heaven's sakes? So many questions. And the big question for me is, how many of the chemicals that we associate with various plants are actually produced by those plants? And how many of them instead are produced by the fungi that are living inside the leaves and twigs of those plants? It's extraordinary to me that every single plant that anyone's looked at has multiple fungi living inside it, everything from the tiniest liverworts to the tallest Douglas fir. So I am going to talk about these 3, but there's lots more. Lichens are beautiful, they're diverse. There are at least several thousand species of lichens that live in British Columbia. They are generally at least 95% fungus. They contain algae and or cyanobacteria, recent research suggests that a lot of our larger lichens are probably at least 2 species of fungi living with at least 2 species of algae and cyanobacteria in some of them. And so it's probably better to think of these organisms as ecosystems rather than as species. I won't talk too much about lichens, but it would be simple enough to do another evening just based on lichens. They're so fascinating. It's amazing to me that a lichen like this lung lichen, this particular species, contains a fungus, an alga, and a cyanobacterium, which is some people call blue-green alga, but it's a type of bacterium that's also photosynthetic, that a lichen like this that contains 3 of the major kingdoms of life on Earth we think of as just one organism. Absolutely remarkable. And the lengths that the fungi go to cultivate the algae are extraordinary in a lot of cases. One of the great plant fungal partnerships, lichens. It allows the fungi and the algae to live in places where neither of them could live on their own. Mycorrhizae. More than 90% of, Mycorrhiza means fungus root, and Mycorrhizae is simply the plural. And it recognizes the fact that more than 90% of vascular plant species on Earth have fungi growing associated with their roots, right? And we should also recognize, I think, that the oldest fossils that people have found of land plants that are about 400 million years old or so have fungi growing, have fossilized fungi growing with the fossilized roots. And it may very well be what allowed plants to climb out of the water onto land, the fact that they had fungi associated with their roots. Where we see plants that have fungi associated with their roots and where people have done good research on it. So remember, this is more than 90%. Almost all vascular plants have these fungi growing associated with the roots. It's what is called an obligate relationship. That if you kill the plant, the fungus will die. And if you kill the fungus, the plant will either stop growing or in most cases die. Okay, so it's an obligate relationship. They need each other. There are different kinds of mycorrhizae, and the fungi involved have different kinds of fruiting bodies. But I told you before that most of the small mushrooms that we see in the woods are the fruiting bodies of decomposer fungi. Almost all of the medium and large-sized mushrooms that we see in the woods in British Columbia are the fruiting bodies of fungi growing attached to the roots of trees. The implication is, and I think a lot of any mushroom foragers here tonight will appreciate this, if you cut down the trees, the mushrooms will disappear. What's not so well appreciated, because it's really impossible to do it in the field, but it's been done in the laboratory in controlled experiments. If you were somehow to get rid of the fungi, the trees would disappear. So when we talk about the importance of fungi in forest ecology, I think you have to start from an appreciation that if we didn't have the fungi, there wouldn't be any trees and there wouldn't be any forests. That's rather important. Imagine Salt Spring Island or British Columbia without forests. And those forests exist because of the fungi. There's different kinds of mycorrhizae. And the ones that I think are of most interest tonight are what are called Ectomycorrhizae. These different sorts, there's probably a dozen different sorts, defined by the nature of the association between the fungus and the root and the fungi, the type of fungi that are involved in the partnership. Most plants on Earth form arbuscular mycorrhizae in the top right there. And most plants in the heath family or heather family in BC form ericoid mycorrhizae in the lower left there. But neither of the fungi involved in neither of those two types of mycorrhizae form macroscopic fruiting bodies. Neither of them form fruiting bodies that we can see without a microscope. On the other hand, the Ectomycorrhizae in the top left hand there, which is a relationship we only see in about 3% of vascular plant species globally, but we see it in the pine family, we see it in the birch family, we see it in the families which are important from the point of view of the trees of British Columbia. And so Ectomycorrhizae are only on 3% of plants globally, but in British Columbia, they are the type of mycorrhizae that we find in our trees, in our forests. And the other thing to keep in mind is that Ectomycorrhizae, more often than not, form fruiting bodies which are mushrooms. They form fruiting bodies we can see. And that's why the Ectomycorrhizae are important for the forests of British Columbia and important for the mushrooms of British Columbia. I talked about how a plant has its mycorrhizal fungi associated with its roots. The work of Suzanne Simard and others is the best demonstration in north temperate forests, really, of the complex nature of this relationship. We did a study of the mycorrhizal relationships of forests in the Victoria watershed. Each of the big old Douglas fir there had between 12 and 14 different species of fungi growing with its roots. So it's not a tree and its fungus. In the case of the Victoria watershed, it was a tree and its maybe 14 different species of fungi, and those fungi are all attached to other trees. And what you get instead of a tree and its fungus is you get a network which connects every tree in that forest with every other tree in the forest, what's sometimes called a common mycorrhizal network or a wood wide web. And those trees are exchanging water, they're exchanging sugars, they're exchanging minerals, they are exchanging signaling hormones that say things like, hey, I'm being attacked by a bark beetle, raise your defenses. These are incredibly complex systems that probably depend a lot more on collaboration and cooperation than on what the textbooks would have you believe, which is out and out competition. Fascinating stuff, cutting edge science. There's lots of books and movies about it right now, but it's fabulous stuff. And I'll just skip that because I do want to stay roughly on time here. And I want to talk briefly about Mycoheterotrophs, and then I have a few closing remarks. So Mycoheterotrophs, we'll skip that. Mycoheterotrophs are vascular plants that lack chlorophyll. And they tend to catch your eye. They're the coral root orchids, they're the pine saps, they're the ghost pipes, as in the left-hand picture here. And most books will probably tell you that these plants derive their nutrition from decaying organic matter. They're saprophytes. And in fact, it's way more interesting than that. The ghost pipes, like more than 90% of vascular plant species on Earth, have fungi associated with their roots. In British Columbia, the ghost pipes have the fungus that makes the short-stemmed russula mushrooms attached to their roots, and that's the only one that's been found so far. So you have the mycelium of the short-stemmed russula fungus attached to the roots of the ghost pipe, but it's also attached to the roots of nearby conifer trees, usually Douglas fir. And what that fungus does, in addition to making the short-stemmed russula fruiting bodies in the autumn, is it takes the sugars that it derives from the roots of the Douglas fir tree, moves them through its mycelial network in the soil, and feeds the ghost pipe plants. And that's how the ghost pipe derives its nutrition. I think a lot more interesting than simply being a saprophyte. Another example, which only works in southwestern British Columbia, because that's the only place we find candy stick. Candy stick is another mycoheterotroph, or another one of these vascular plants that lacks chlorophyll, so it can't make its own food. And the other effect it has, of course, is it makes it look distinctively weird in the forest. You're not used to seeing plants that aren't green. The mycoheterotroph, the candy stick, the only fungus that's ever been found attached to the roots of candy stick is the fungus whose fruiting bodies are pine mushrooms. So if you see candy sticks fruiting someplace, flowering or fruiting someplace in the springtime, and you go back in the autumn, in all likelihood, you will find pine mushrooms. That's useful information. Even if nothing else in the whole talk tonight is useful, there's a way to hunt for pine mushrooms. Okay, a few closing remarks. The best study of mushrooms and habitats was done in Clayoquot Sound. And they sampled, some of BC's best fungal scientists sampled 6 habitats: dunes, spruce fringe, old growth forests, second growth forests, bogs, and estuaries from 1997 to 2001. They collected 551 species of macrofungi, think mushrooms. 17 to 36% of the species in a habitat were found only in that habitat. So if you want to maintain species diversity, maintain all of your habitats. Of the 551 species, only 28 were found every year, and 308 were found only in 1 year. Mushrooms don't fruit every year. If you want to do an inventory of them, you need a commitment to a multi-year survey. And finally, old growth forest had approximately 4 times as many species as bog and estuarine habitats, and 1.5 times as many species as the rest. Old growth forests are important. If you needed one more reason to understand why old growth forests are important, add mushrooms to the list. Kaufman's root shank in the lower right-hand corner there is a mushroom species that grows exclusively or almost exclusively in old growth forests. We're not sure why, but we don't find it outside of those habitats. If you want to maintain your fungal diversity, you have to maintain some old growth forests. Again, add it to the list of reasons why that's important. The only other thing I'll mention about fungi and forest ecology is the effects of a changing climate. Our climate's been changing for quite a while now. Some of the things we've been seeing that you've been seeing on Salt Spring Island are tree death, which can be caused by drought stress, but it is sometimes also caused by the effects of fungi attacking trees that are otherwise stressed, which is the drought stress can kill some trees on its own, but it can also render the trees a lot more susceptible to a whole host of fungal ailments that can come in and finish it off, including a lot of these parasites that under normal conditions, the tree would be able to live in a somewhat harmonious relationship with. But if we're seeing increased summertime drought, we're going to see the sorts of effects we've seen involving fungi and arbutus, the leaf blights, including some Douglas fir trees as well. We'll see changes in incidence, extent, and severity of forest fire. Remember that an awful lot of almost all of the medium and large-sized mushrooms that we see in the forest are the fruiting bodies of fungi associated with the roots of trees. If the trees are killed, then the fungi die as well. So forest fire affects those fungi. However, it also creates much larger burned areas than we've ever seen before historically in British Columbia. And there are mushrooms we call phenocoid fungi, which react particularly well to the burning of forests. And I've included pictures of a couple of them at the bottom there, the Geopixus, a cup fungus that we just about always find on burned areas. And of course, some of the species of morels like Morchella tomentosa that are picked commercially in British Columbia. The morel pickers who come out in the springtime following a big forest fire year, the successful ones at least have forest fire maps with them and they know where to pick. So some fungi will do better following an increase in incident, extent, and severity of forest fires. Around a lot of, at least the southern part of the province, the tree line is increasing in elevation. That is, the height to which trees can grow is increasing in elevation. And as a consequence, the area above tree line, what we call the alpine, is shrinking. And there are a number of species which are mycorrhizal species of things like dryas species and dwarf willows and plants like that that grow in the alpine. And as the alpine disappears, we can expect that they will disappear. At the same time, the fungi that grow with the roots of trees will expand their range as trees expand into the alpine. And finally, we're starting to see some additional fungal species moving north. And as we're seeing plant species moving north as the habitat becomes a little more hospitable. And that's just something to keep our eyes on. Thanks.
Okay, great. So that was incredibly interesting, Andy. And I do see a couple of questions in the chat box, but if anybody else has questions, please type them in now. So here's a question from David Denning. He says, what is the animal that carnivore oyster mushrooms are feeding on?
Oh, they're feeding on bacteria, yeast, and amoebae. So the animal would be the amoebae.
All right. Okay.
A very small animal.
Right. Okay. Next question is from Vitica Verhoeven. How long can macro fungi live?
Well, it probably varies very much with the species. The world's largest organism, for example, is a honey mushroom in Oregon, which covers hundreds of acres, and so is of course probably millions of times larger than a blue whale. And it's almost certainly been growing for 1,000 years or more. There's nothing that inherently limits the lifespan of a lot of these macro fungi. And I was talking about a lot of the things that grow attached to the roots of trees and produce mushrooms. And certainly the individual trees die, but usually that fungus is attached to the roots of multiple trees. And that's almost certainly a survival mechanism to deal with the fact that that one of its hosts will likely die, but or one of its partners. But if it's attached to enough trees, then it should have another partner that's still alive. So it potentially, unless someone comes along and cuts down one of those forest stands, it could live for thousands of years.
Okay. Chloe asks, any recommendations for a good mycology microscope?
Well, you know, for getting started, a good hand lens is really the tool you want. Dissecting microscopes will allow you to see a lot of the fine detail that most field guides and keys called for. And it's only if you're getting into some of the more difficult groups like Mycenas and Cortinarius that you'll need a compound microscope if we're looking at things like spore size and cystidia and things like that. We've managed to, our Mycological Society, the South Vancouver Island Mycological Society, has a couple of good mushrooms, or good microscopes, and they've been donated, but we've actually managed to inherit a few from school systems, so that's where I might start. There was a shop in Vancouver, I'm not sure if it's still operating, that sold optical equipment. So you might check that out too. Sorry, that's not a great answer, but
All right, Dave Secord asks you, what can you tell us about marine or estuarine fungi? Do they exist in what kinds of habitats or on what hosts?
one of the very few fungi that I actually described was a fungus that decays racweed, fucus species. So there are not a lot of marine and estuarine fungi. They don't handle salt water particularly well for the most part. There are fungi that grow inside marine animals. There are a number of, once you get into estuaries, there's a lot more fungi because they'll do well in brackish water or fresh water, but out in the open ocean, there aren't an awful lot.
All right, Charlie asks, how do you find and study those mycorrhizal connections between the firs? Does it involve a lot of digging? Or what?
Yes, yeah. So one of the reasons that Suzanne and students and even some projects I've been involved with have been making such great headway in the last decade in trying to understand these systems is because of the development of these new technologies. A lot of it has to do with DNA. So if you can extract the fungi from the roots, so we'll sample the roots and fungi in the soil, bring them back to the laboratory, separate the roots from the fungi, and then extract and amplify the DNA from the fungi, then you can identify them to species or genus, depending on whether the sequences are already in GenBank. And it's the sort of the development of the relatively rapid, inexpensive DNA technologies that's allowed that kind of things. So let me just share here again. I'll show you one picture. Let me see here. I'll go. Let's go back here a bit. In this study, I kind of skipped over it because I didn't want to get into the details here. They were looking at the connections. So this is a 50 meter by 50 meters square, the square in southern interior British Columbia. The green items are Douglas fir trees. The big green items are bigger Douglas fir trees, bigger older trees. The lines are two species of rhizopogon, one blue and one pink. Rhizopogon are mycorrhizal fungi that form truffles as their fruiting bodies. And so they would sample the root they would sample all the fungi growing with the roots of one tree and then all the fungi growing with the roots of another tree. And if they found the same fungus growing on the roots of two trees, they would draw a line between it. So these lines represent the connections between the trees in the stand of two species of rhizopogon. Now we were finding maybe 6 or 7 times that many species on each tree. This is just looking at 2 species. So multiply the number of lines there by 6 or 7 times. And the work was done by sampling the roots of each tree and looking at the fungi that were growing there. And if they found the same fungus growing on another tree, they would draw a line between it, the same individual. So that's how that work was done. And that's just an example of how you try and understand these systems, really up until the development of inexpensive, rapid, relatively easy to use DNA technology. You can imagine people trying to sort out what's happening with microscopic threads running 30 centimeters below the soil in a forested stand. It's estimated, for example, that in 1 thimbleful of forest soil, there are 2 kilometers of hyphae.
Wow.
So it's an amazing system, and some of the work, like some of Suzanne's early work, was done in the laboratory, and some of the detailed stuff can only be done in the laboratory, where she would, for example, for her PhD, which was a really fun project, she took a Douglas fir tree and a paper birch tree and had them both hooked up with the same mycorrhizal fungus in her labs at Oregon State University. And she would shade the Douglas fir and document the transfer of sugars from the paper birch to the Douglas fir, or shade the paper birch and document the transfer of sugars from the Douglas fir to the paper birch.
Wow.
That's not something you could do in the field, but she was able to do it in the laboratory. And I remember asking her what possible benefit there could be for the paper birch in donating some of its sugars to the Douglas fir. And I remember she rather shook her head as if I was the biggest idiot in the whole world. And she says, why do you assume that the plants are in charge? You know, imagine you're a fungus and your survival depends upon being hooked up to the roots of a living tree. So you attach yourself to multiple trees, of course, and then try and keep them all alive, right? It's a different way of thinking about the world, really.
All right. So Mart asks, why are fungi so speciose compared with birds?
Gosh, that's a good reason. I can't say I've ever thought about that. It could be in part because they do a lot of different, they do a lot more things than birds. I mean, birds just kind of fly around and eat shit, don't they? Sorry, I'm teasing. I know any group like this has lots of birders. But fungi really have a whole lot of different niches out there, and they form all kinds of different partnerships. I framed the whole talk tonight in terms of all the different things that fungi are doing out there in the ecosystems. So maybe that just provides a lot more opportunities for them. But then I think about it, that's not a particularly good argument, because plants are largely all doing pretty much the same thing. They're all autotrophs. They're all making their own food, and they're much more speciose than birds too. So I don't know. That's a good question. How come birds are so depauperate in species? There we go.
Well, I'm not going to answer that one. So we'll move on to Jane's question, which is, meadow mushrooms seem to be found in pastures. How does this work where there are no trees?
Meadow mushrooms are the fruiting bodies of decomposer fungi. And so they don't have to grow with where there are trees. But even things that grow with trees can grow sometimes more than twice the canopy width of the tree away from the tree. They don't have to be right in close to the tree. And that makes it really complex. For example, I'm involved in a study right now that's looking at the mycorrhizal fungi associated with the roots of Gary oak, which we know nothing about in British Columbia, and seems to be full of undescribed species as far as we can tell from the DNA. And you have to be sure that there's not a Douglas fir usually within 100 meters of the Garry Oak, or else you run the risk of, in your sampling, picking up the fungi associated with the roots of Douglas fir when you're sampling for Garry Oak.
All
But,
right.
yeah.
Okay, Molly asks, what is the smallest fungus in the world?
Oh, well, I imagine it's the one that you make bread with, Molly, or you brew your beer with. It would probably be a single-celled yeast.
Okay. Emily asks, what are some good ways to learn more about mycology? Other than reading your book.
Yeah, I think probably the best way to learn more about fungi is like the best way to learn more about birds or plants or any other group of organisms. That is to get together with people with similar interests. So if someone said, how do I learn birds? I would say, well, go out with birders. You know, to learn about fungi, British Columbia has two mycological societies, two mushroom clubs, the Vancouver Mycological Society and the South Vancouver Island Mycological Society out of Victoria. They both have websites. They both have lots of activities. I'm off this weekend to the South Vancouver Island Mycological Society's annual Cowichan Lake Foray. So there are all those activities. If you're not particularly keen to leave Salt Spring Island, now that Salt Spring Island has a terrific new natural history society, no doubt they'll have lots of fungal events. So that will probably be a good way to move forward. So we generally recommend, if you're close to Vancouver or Victoria, those two mycological societies are full of fungus nuts, including me. I'm the past president of the South Vancouver Island Mycological Society. If you're anywhere else in the province, a lot of the natural history societies have people in them who are keen on fungi, and that's a good place to start anyway.
Great. So Vitica asks, are fungi species stable through time? Have they existed as the same species for millions of years?
I don't know the answer to that. Fungi are poorly represented in the fossil record compared with things that have bones or teeth or anything like that. However, there are, and also the other complicating factor is a lot of, say, fungal mycelium, a lot of hyphae look like each other. So if you showed the world's best mycologist photos of a couple of different types of mycelium, they wouldn't be able to tell you what species they came from. Ditto for a lot of different spore types. But there have been some mushrooms that people have identified to species in amber from tens of millions of years ago at least. So some of them seem to be, but beyond that, I don't know. I don't have a good answer, and I don't know if somebody else would, but it's not as good a fossil record as things that have hard parts.
Okay, Emily again asks, what's your favorite fungi?
Gosh, that's such a tough call. It's different every day. You know, I was in Bamfield at the Bamfield Fungus Fest this year, which is a really fun one to get to. Bamfield's one of my favorite places in BC, and I've got a lot of good friends there. And I go there, and there is one of those cordyceps type fungi now called Tulipocladium that fruits out of deer truffles. And Bamfield seems to be the BC center for it. And every time I go to Bamfield, I find this fungus fruiting out of deer truffles. And that day, that is my favorite fungus. But then other days, things go really bad. Well, and even a day like today, in the fridge in the next room here is a bottle of a very nice Quebec ale. And my favorite fungus after the talk tonight is going to be the strain of Saccharomyces cerevisiae that brewed that bottle of beer that's in the next room.
All right. So why do you, Mart asks, why do you suggest there is other than competition structuring the forest community? The fungus transporting nutrients is stealing from one to give to another, which it benefits from. The competition would be among the fungi themselves, members of the same species, and the adaptation of moving nutrients about is self-serving among others of the same fungus species. No, question mark?
Probably. That's another great way of looking at it. Thank you very much for that. That's actually quite cool. It's not, I mean, you have to start thinking about everything different once you start thinking underground. So most of the textbooks that I grew up with, at least, and some that I've read fairly recently, are still talking as if anything really important happens above ground. And so when they talk about what's happening above ground, that's where the conversations are about competition and winners and losers. Once you realize that most of what's important is happening below the ground, then I guess you have to start thinking again in terms of competition and cooperation. And it may very well depend on whether you're thinking about it from the fungus or the plant point of view. You know, the fungi are no doubt competing against each other. And if the fungus really is running the show, then it's not the plant that is cooperating with other plants by sharing nutrients with it, but it's the fungus that is competing with other plants by moving material from one to another. So perhaps you're quite right. I have to start once again thinking of forests as operating in a fundamentally different way. Good.
Carlos asks, other than not disturbing the forest, is there something that individuals can do to encourage the diversity and extent of the mushroom species?
Sure. Don't log them. Leave the trees standing. A lot of the fungal species in a forested province like BC are associated with forests. And we know that a lot of the fungal species disappear when the forests are logged.
Yeah,
and that applies on an island as well as it does to a province as a whole.
Yeah, I think he's asking, other than not doing that,
Oh,
or anything
okay.
else that you can do. I was wondering about that too. Are there rules of thumb about picking mushrooms, but not?
Well,
yeah.
So remember that the mushrooms are the fruiting bodies of the fungus. Normally we explain it as being like apples on a tree, but you could think of them as berries on a bush. And you should be able to go back and pick the apples from that tree each year and go back the next year and pick apples from it again, so long as you don't break any of the branches. So if you are harvesting mushrooms, in theory, you should be able to harvest the mushrooms and come back and harvest them the next year, so long as you don't dig up the soil while you're digging up the mushrooms. This is actually borne out in the only good study that's been done in this part of the world in the last few decades, and that was done by the Puget Sound Mycological Society that did an experiment over 5 years where they established one patch of land where they picked chanterelles, another patch of land where they cut off the chanterelles at ground level, and another patch of land where they just left the chanterelles. They didn't pick them at all. And they found no difference in productivity in those 3 areas in future years. So both from a theoretical point of view and from an experimental point of view, though granted only one small citizen science experiment, it seems that if you don't destroy the place, the mushroom should be all right. It's, in fact, probably even a more robust system than foraging for wild plants, because the mushrooms produce an extraordinary number of spores. So a chanterelle mushroom, for example, would probably produce hundreds of thousands of spores, at least, perhaps millions of spores, one mushroom. Which means that even if people go in and heavily harvest an area of its chanterelles, if they leave 5% of the chanterelles behind, that should still produce tens or hundreds of millions of spores per hectare.
Okay, great. Well, we're at 8:30 now, so we've pretty much run out of time. So I'm going to ask Deborah if she can save the chat. And would you be willing to answer some of these after by email or something, Andy? I don't know if that would work. That may not work.
No, that's what Richard and I did following our seminar earlier this year.
Okay.
And I'd be happy to do it again, absolutely.
Okay, thank you. That would be great. So Deborah, if you could do that. And sorry, people, it's just a testament to the interest here that we've got so many questions. So anyway, I'd like to ask David Denning now to thank Andy for his talk tonight.
Andy, that was really great. Thank you so much on behalf of both our Salt Spring Naturalist group and the Conservancy. It's really amazing to hear you speaking about something you have such passion about, and it really is catching. I learned a couple of things. For one, that really the great biodiversity that we have here in our forests is really associated with cooperation. And I think this tells us a lot. If there's a lesson to be learned, and more metaphorically perhaps than written in the science, but if there's a lesson to be learned, cooperating together and working towards preserving nature and making it regenerating where we can is, in the spirit of fungi, is certainly something we should all try to do. And I'm looking forward to having you involved with that more. In fact, it seems to me you've committed to coming to Salt Spring and giving us a walk through the forest. And I would imagine that some of that would be fungi stuff, but also I'm betting we could get you for some of the great plants that you've identified in all your different guidebooks that you have. So we're going to get you as soon as we can. COVID is hopefully being left behind enough that we can get you here, Andy. So with that, I would have to say that today you are our favorite fungus expert, and I think you will remain more so over the time. So thank you again, Andy, and come back soon.
I will do that, David. Thanks so much.
Okay, great. So that ends our presentation tonight. And again, thanks, Andy. And thanks to all of you who participated. Night.
Good night.
You okay with the saving the chat, Deborah?
Yep, I've already saved it, so we're good.
All right.
Thank you.
I'm gonna, should we stop the meeting then, or do you want to wait till these messages stop?
I think we're okay to stop now.
Okay.
Okay, thanks, Susan.
Thank you.
Good night.
Bye.